A year in Principles of Biomedical Science. At the beginning of the year, we made videos to demonstrate the different crime scene search methods to show our understanding of how to investigate for Ana Garcia's crime scene. There are 6 different methods for this project: zone, spiral, line, wheel array, link, and grid methods. One of our next projects was our blood spatter experiment. We had to look at the patterns of blood when spattered at different heights. We then had to measure the distance of the farthest spatter from the middle to see how far the spatters had reached. For the strawberry DNA experiment, we took strawberries and combined them with an extraction liquid made of water, salt, and dish soap. We added rubbing alcohol, which allowed the DNA to be scooped out and extracted from the solution. For the heart dissection, we used sheep's hearts, which are very similar to human hearts, to weave through string to represent the pathways that oxygenated and deoxygenated blood use to go through the heart. We used blue string for deoxygenated, and we used red for oxygenated. For our midterm project, we had to design an original board game that quizzed players about the heart and blood flow. We made questions on the anatomy, different heart surgeries, and blood types, and we had to make the cards and pieces ourselves. When we did the phlebotomy lab, we made a video showing how to properly take someone's blood. We used red dye, a fake arm, and a needle to demonstrate this. It was to show our understanding on the topic and how we were able to teach others. During the weightlifting experiment, we measured in groups how the length of time between sets of lifting weights affected the difficulty to lift the weights and the human heart rate. We used this information to determine a good wait time between sets. For the patient 0 experiment, we were all given test tubes with water, besides for one person who had a chemical indicator that would change color when triggered. We then put some of our solution in 5 others' tubes, and at the end we triggered the color change to see who had the virus. We then had to trace back to find patient 0. For the bacterial inoculation lab, we made videos like the one shown demonstrating how to properly perform a bacterial inoculation on an agar plate. We showed how to properly and safely spread the bacteria and how to put it in to incubate. At the very end of the year, we performed gram staining on gram-negative bacteria. This means that at the end of the process, the bacteria should be the color pink. We had to individually perform all the steps correctly without messing up or skipping a step, and at the end were evaluated by Mr. Sharp on how we did.